In the real world of genetics, we often have multiple alleles. So there are many different versions of a gene, and we call these different versions alleles. Sound confusing? Let's look at codominance in the blood groups. There's A, there's B, and there's O alleles. None of them are dominant. Each of them codes for a different surface polysaccharide on the red blood cells. And this gives us our own personal chemical identity that helps us to identify self from non-self. Why do we care about non-self? Well, we don't want to be invaded and destroyed by some bacteria, and that's why we have these different markers and different blood groups. But it becomes a problem when we try to transfuse blood. So the genotype AA, which is a phenotype or expression of a A blood, can't be transfused into a B blood because they're different markers. Now if you're O, you have no surface markers for this specific trait. And I'll talk about that, why it's important, in the next slide. AB has both A and B surface markers, and so it's going to recognize all blood types. It's the best recipient for an emergency transfusion. O doesn't receive blood very well, except its own type, O. However, it is a preferred donor in emergency situations because it lacks surface markers for this trait, and so other blood types are unlikely to attack it. Incomplete dominance, as the name suggests, is when there's an intermediate of the alleles. There's no dominant. Flower color, if we cross a red and a white flower, we're going to get a pink. What if we cross a pink with a pink? Any idea what the results will be? Well, here it is. The phenotypic ratio, all that means is the expression of the genes. Remember, phenotype is physical expression, is 1 to 1. 1 white, 2 pink, 1 red. Classic incomplete dominance cross. In polygenic traits, there may be several genes that are going to combine to produce a phenotype. Example would be skin and eye color. Some people have very dark skin and some have very light skin. And all this is determined by the genes that code for the production of melanin. It's a brown pigment. The more you have of these genes, the darker your skin. Now, why do eyes sometimes look green or gray? It's because of the reflection of light. But all skin and eye color and hair are all coded by genes that cause the production of the brown pigment melanin. In sex-linked traits, I know this sounds exciting, but it's really kind of depressing for me. The Y chromosome is very small. It contains relatively few functional genes. This means that whatever is on its partner, remember genes occur in pairs, whatever's on the X chromosome is going to be expressed. That could be a problem because what if an undesirable recessive trait is on the X? There's no backup. I said this is depressing because this is the condition that all men experience. We all have the Y. The Y chromosome accounts for many of our genetic problems, and it makes women genetically superior to men because the women have a backup. Take a look at that lower right diagram I made. XX. That means you have to have 2 recessives to suffer from something. But in the XY, 1 is enough to cause a problem. Now women may carry undesirable traits, and we use that word carrier, but they're still normal. They have the normal phenotype. Example with hair follicle mass reduction, it starts to cycle down in men in response to our production of DHT, dihydrotestosterone. Now, not all men are susceptible to this, but eventually, most of us are. I mentioned the concept of a carrier. So a woman has normal vision, but she carries the gene. What if she marries a man with totally normal vision? Well, if he has normal vision, then he has a normal gene for this trait. But the woman's a carrier. She's going to have dominant and recessive. So what is going to happen, I wonder, if they were to have children? Do their daughters and do their sons have an equal chance of being color deficient? If you recall, during meiosis, the gene pairs are going to separate. And that is why I set up a box like I have on the right side. Hey, can you read this number? If you can't, then you're probably a man. Here's a solution. It may look confusing, but think back. X and Y, that's a male. XX is a female. So there's the dad on the top. He's an X, big N, Y, nothing. Remember, the Y is really tiny. His gametes are going to join in a random way, and that's why we set this problem up. If you look at the far right boxes, there are the two boys. X big N, Y, X little n, Y. So what's the chance of a son being color deficient? 50%. Now sometimes on a test a student will write 25% because they put in every box. 1 out of 4. But I'm not talking about the girls in that question. In the case of the girls, because this is sex linked, it's a little different than the other genetics problems. Because here we actually separate out the sexes because they are going to respond differently. And in this case, all girls have normal vision. Half of them are carriers. Penetrance refers to the probability that if we have the allele for a trait, that we'll actually suffer the phenotype, the physical expression. We have many recessive genes in our chromosomes, many. And most of them are not ever expressed. Or maybe they're expressed as we get much older and those genes start to turn on. In a penetrance condition, that means if you have it in a homozygous condition, that means no dominant present. If you have it, then it's going to be expressed. That's 100%. But if you have only, say, 10% penetrance, that means you might have a homozygous condition and those genes are not expressed. And that's pretty cool when it happens. In the case of cystic fibrosis, 100% penetrant. Chloride ions are not transported across our membranes efficiently, and then mucus starts to accumulate in many of the tissues. And one of the problem areas is the lungs, and so they're going to have frequent respiratory infections. However, the medical treatment for this disorder is really improved recently. In aneuploidy, we have a problem that arises during meiosis. If you recall, meiosis is cell division, and if all goes well, we're supposed to have a diploid 2N condition for all our chromosomes. But if there's a mistake, in this case it's called a non-disjunction, that means the chromosomes don't separate when they're supposed to, and we end up with an extra, or sometimes we end up with one less than we need. And that can be a problem. And here's how it works in the case of Down syndrome. On chromosome number 21, if the gametes don't separate properly, look on the right. Hey, we're supposed to divide evenly, but one gamete got an extra chromosome. And when that joins with the partner in fertilization, we now have a trisomy or extra chromosome. Usually not a good thing, although there are some conditions where it's a pretty mild result. In some cases it's fatal though. To detect aneuploidy is not terribly difficult because we're dealing with an entire chromosome abnormality, not just a gene. We start with fetoscopy, and here we're going to use ultrasound that's recording the echoes from the fetus, and we do that so that we can always monitor the location of that fetus so that it's not punctured when we insert the amniotic needle. We're pulling out the skin cells of a fetus. Just like you and I, we're constantly shedding skin cells, so does the fetus. Those are drawn into the hypodermic needle, and the cells are cultured using chemicals to stimulate the cells to grow and divide so that we can examine them microscopically and determine if there is a problem. On to the karyotype, where we photograph these chromosomes as they appear through a microscope. We add some dye so that they're visible. We arrange them from largest to smallest, and we examine them for aneuploidy. Is this a boy or girl? Well, if you check out the X and Y, that'll answer your question. Do you see aneuploidy? I can see it. Gene activation. Genes just stop functioning. We experience this often as we age. There could be environmental factors. Cigarette smoking will inactivate genes, but so will just simple aging. In the case of lactose intolerance, lactose is one of the milk sugars. We need an enzyme called lactase to hydrolyze or break up milk. As some people get older, that gene coding for lactase becomes inactive, and then milk sugars begin to build up. The person feels tired, bloated, headachy, other symptoms. In some cases, pretty mild, but maybe quite severe. A point mutation is an insertion or deletion of a single gene. In most cases, it's not a problem. All of us have point mutations, and we're not even aware of them. However, if we want to screen a mother, well, I should say a pregnant woman, then we can do an alpha-fetal protein test by doing a venipuncture. We draw blood from the vein, look at that blood sample, and get some information about the liver of the fetus or the activity of the amniotic sac in searching for a AFP problem. There are some serious conditions, such as spina bifida, which the AFP test can detect. And this is good because if we know spina bifida is going to be a problem, we can have a surgeon on hand. So then when that child is born, the problem can be addressed quite quickly, and recovery is that much faster. Summary here, multiple alleles, that is, different versions of a gene are called alleles. Codominance is where no gene is dominant, and so we end up with different alleles like A, B, O. They're going to code for the polysaccharide surface markers on our red blood cells. AI or AA, think of A blood. So we have genotypes and we have phenotypes. AB is the best recipient because it recognizes everything. O is the best donor because it doesn't have these surface markers. Incomplete dominance, that's where we get a white and a red and a pink flower resulting. Polygenic, there's the word poly, many genes, such as the genes that code for melanin. The more genes that code for melanin, the darker your skin and eyes and hair. It comes down to just lots of genes. Sex-linked traits. If you have a Y chromosome, it's rather sad because that means you have few functional genes on that, and that means there's no backup. So a recessive on the 1X is what you're going to express, whether it's good or bad. GHT, that's one that causes the hair mass to cycle down and get thinner. Penetrance, that is the likelihood that a gene is going to be expressed in the phenotype. And cystic fibrosis, unfortunately, is 100% penetrant, meaning that if you have a recessive homozygous condition, then you got it, man. You can't dodge this bullet. And that's where chloride ions are not transported properly, and there's mucus accumulating. Aneuploidy, it's an error usually during meiosis where the chromosomes don't separate as they should, a non-disjunction it's called. Down syndrome is one example where we end up with a trisomy of number 21. We can test for aneuploidy with a fetoscopy where we use ultrasound and a very long needle to extract fetal cells through amniocentesis. We're going to culture those cells and photograph them through a process called karyotyping and look for extra chromosomes or sometimes missing chromosomes. Gene inactivation, think of lactose, the milk sugar that is broken down by lactase through hydrolysis. And as we age, or sometimes just certain cultures don't tend to process milk very well. Finally, point mutations or where a single gene is deleted or inserted and we have conditions. We can test for this with a venipuncture and maybe detect something like spina bifida. Genetics is wow.